Hello, 2018 Phage Hunters. If you work across the room and I've managed not to talk to you yet this semester, my name is Ben, and I'm from Pennsylvania. Today I would like to introduce you to the bacteriophage that I've worked this semester to isolate and characterize. My bacteriophage is named after my friend and former Science Olympiad partner for the event Microbe Mission, Lauren. While studying for the aforementioned competition, I came across the phage hunting program before I had even been accepted to JHU. During this program, we were required to know some of the basics about phages and their replication, such as the lytic and lysogenic cycles. Anyway, we came across the sea-phages program, and realized that both of the schools that we thought we would be attending offered the Phage Hunters program for freshmen. Because we both had an interest in microbiology, we decided that we would enroll in the program and compete to find the best phage. Unfortunately, because Lauren decided to attend the University of Delaware, which doesn't offer the Phage Hunters program, we were unable to both take the class at the same time. Out of respect for her, I decided that I should name my phage Lauren, after her. So class, let me introduce you to microbacteriophage Lauren.
Hello everyone, my name is Lauren. I was collected in a flower bed outside the FFC on September 26th at 2 p.m. and 71 degrees Fahrenheit. Ben had a bit of trouble finding a phage and actually tried 5 different dirt samples before finding me where he did. Right when he was about to give up and adopt someone else's phage, he found me and he definitely picked a winner.
Wow, thank you, Lauren. Now to talk about the process that got me to where I currently am in the process of characterizing my phage. The first plaque that I got in Phage Hunters was the result of an enriched isolation of the dirt sample that I collected in the location mentioned earlier. As Lauren said, it took me a few weeks to collect a viable sample. However, the most frustrating part of my phage discovering process was actually getting a spot test to work for me. I was caught in the midst of the contamination debacle that affected many of us. Because of the contaminated bacteria that we were plating with, it took me 3 tries, meaning 3 weeks, to get a successful spot test for my original plaque. After I conquered the spot test, I moved on to the process of performing serial dilutions. However, instead of doing the 3 required dilutions, I accidentally got caught up in the craziness of midterm season and performed a 4th serial dilution for no reason. But because of this, there is no doubt in my mind that my phage population is pure. As for the morphology of my pure phage population, the plaques created by this phage have cloudy or turbid margins, indicating that the phage itself is likely temperate rather than lytic. The size of the plaques decreases with the quantity of plaque-forming units, as higher dilutions generally result in larger plaques. Now wasn't that refreshing? Now, back to the presentation. The titer I've collected of Lauren particles is actually an extremely high concentration of 3.8 times 10 to the 11 PFU per milliliters, which was collected directly from a web plate with no extra lysate concentration steps needed. I also have 2 backup tubes of lysate collected with an unknown titer, bringing my total volume of lysate to around 16 milliliters. Under gel electrophoresis, a bright white band appeared under both of my samples, indicating a high concentration of genetic material present for sequencing of around 100 nanograms per microliter. During spectrophotometry, I had a couple of curious results, but after talking with Dr. Fisher and reviewing the results of the electrophoresis, I believe the concentration of my DNA is more than likely high enough to be sequenced. Assuming we started with 78.8 nanograms per microliter, I should have more than enough DNA for sequencing. Finally, we get to one of the most interesting parts of the phage characterization process, the images obtained under EM of Lauren. So, yeah, look at these cool images. She's pretty cool looking, isn't she? Note the tail size, capsid, girth. Yeah. She has a long tail. Yeah. So, Lauren, why don't you finish this presentation for me?
In conclusion, I am an extremely important, cool, and interesting phage. Ben has collected for next semester a very high concentration of lysate with plenty to spare, a genome already prepared for sequencing courtesy of Dr. Fisher, and a phage with a weirdly sentimental backstory. What more could you want? I would be honored to represent the phage population of the 2018 phage hunters by allowing you all to annotate my genome. Thank you.